Chapter 1: The Microbial World and You. What is microbiology? Microbiology is the study of microorganisms, also known as microbes and viruses. What are microorganisms? Microorganisms and viruses are generally too small to be seen with the naked eye. Microbes versus a germ. A microbe, again, is an organism that are usually too small for us to see without a microscope. However, a germ refers to a rapidly growing cell. So usually try to use the word microbe and germ interchangeably, but in reality they mean two different things. A microbe is an organism that's too small to be seen with the unaided eye, and a germ refers to a cell that is growing quite rapidly. In spite of their tiny size, microbes have an enormous impact. They maintain our environment in life-sustaining balance, and they are some of our most valuable industrial tools. If they are left unchecked, they can destroy our livestock and crops, and they can spoil our food. A few of them can make us sick and even kill us. In this chapter, we're going to look at the many microbes that affect our lives. A few microbes are pathogenic. Pathogenic means disease-causing. Microbes decompose organic waste, are producers in the ecosystem by photosynthesis, produce industrial chemicals such as ethanol and acetone, produce fermented foods such as vinegar, cheese, and bread. And they're involved in producing products used in manufacturing, for example, cellulase and in disease treatment in regards to making insulin. Your textbook talks about a E. coli bacteria that produces indigo, and that indigo is produced from a amino acid called tryptophan, and this is actually what they use to put in our genes, our blue genes, to give it that color. Why do we care to know about the microbes in our lives? Well, one, we want to have a knowledge of these microorganisms. It also allows the humans to prevent food spoilage and to prevent disease occurrence. All of the above have led to aseptic techniques to prevent contamination in medicine and in microbiology laboratories. Naming and classifying microorganisms. Carl Linus was the father of scientific nomenclature. He established the system of scientific nomenclature, which was the means of communicating to others about specific organisms. The classification of microorganisms is a binomial system. Each organism has two names, the genus and specific epithet. The genus is the genera. It's the first name, which is always capitalized and underlined or italicized. The species or the specific epithet is the second name, which is underlined or italicized, but not capitalized. The names are Latinized and used worldwide. So if you're good with Latin, then this is a breeze for you. And they may be a descriptive, like describing the organism, or they may honor a scientist. So let's take a look at some scientific names. Staphylococcus aureus. Staphylo, if we look at the word staphylo, describes a grape-like cluster arrangement of spheres, which is spherical cocci. So it's saying that when I look at this under a microscope, I would notice that these cells are arranged in a cluster. And you'll notice that each one of these are a circle. So the circles are the shapes, and then you'll notice how they're arranged in a cluster. Aureus describes the gold-colored colonies. So that's what Aureus stands for. And here in this picture, this beautiful streak plate, you can actually see the golden colonies that's produced by Staphylococcus aureus. Escherichia coli, E. coli, honors the discoverer Theodore Escherichia. So here is a picture of him coming up. And the coli describes the bacterium's habitat, the large intestine or the colon. So in your book, Table 1.1 also shows you other examples of some scientific names. So if we take a look at Streptococcus pyogenes, we already know what coccus means. What does coccus mean? If you said spheres, you were correct. So now we know that in Staphylococcus aureus, it was saying that they were arranged in a cluster. Here in Streptococcus, they're arranged in chains. Okay, so Streptococcus, so those circles or spheres are arranged in a chain-like fashion. Pyo means pus, so it means that this particular organism causes the formation of pus. And then down here, they show you Trypanosoma cruzi. In Trypanosoma cruzi, you'll see that the cruzi came from honoring the epidemiologist that discovered it, Oswaldo Cruz. After the first use, scientific names may be abbreviated with the first letter of the genus and the specific epithet. Escherichia coli and Staphylococcus aureus are found in the human body. So I wrote out the entire name for both bacteria. Now, from now on, I will be abbreviating them. So E. coli is found in the large intestine, and Staph aureus is on the skin. So now that we went over what the scientific name is, which of these is a correct scientific name? If you chose the letter C, you are correct. Classification of microorganisms. Classification allows us to arrange organisms into related groups. In 1978, Carl Woese came up with a classification system for microorganisms. So he classified them by cell types. The classification system groups the organisms into 3 domains: bacteria, archaea, eukarya. Eukarya consists of protozoa, fungi, algae, and animals. So in microorganisms, we're talking about, or microbiology, we're talking about multicellular animal parasites. Viruses are not included in the 3 domains, and we will discuss that in a few moments. This is just a phylogenetic tree showing you the 3 domains. Bacteria. Bacteria are prokaryotes. Prokaryotes means before the nucleus. So that means that these bacterial cells do not have a nucleus. There's no membrane-bound nucleus. Bacteria are unicellular microorganisms. So if you took a look at this picture, beautiful picture, you'll see that this is one organism, another, 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 another. They're unicellular. They have different shapes. So we just talked about coccus. These are rod shaped, so this would be called bacillus. They have what we call peptidoglycan cell walls. They reproduce by binary fission. Some bacteria have flagella for swimming. For energy, they use organic chemicals, inorganic chemicals, or photosynthesis. So that was the first domain. Second domain is archaea. Archaea is also prokaryotic. So what did prokaryotic mean? Before the nucleus. So that means that they have no membrane-bound nucleus. They are also unicellular microorganisms that lack peptidoglycan. They reproduce by binary fission. Archaea usually lives in extreme environments. It will include methanogens, and of course methanogens live in very high levels of methane. Extreme halophiles. They will live in very high levels of salt. Extreme thermophiles. They will live in very high temperatures or very low temperatures. So here's my question for you. Would archaea be considered a pathogen? If you answered no, you are correct. Humans can't live in any of these environments. High methane, high salt, or high temperature or very low, we would not survive. Third domain is eukaryotes, and eukarya stands for true nucleus. So that means that they have distinct membrane-bound nucleus. So their cells contain a membrane-bound nucleus as well as numerous membrane-bound structures called organelles. Eukaryotes are larger than prokaryotes, so their cells are larger than prokaryotes, and they do live in moderate environments. So this is the third domain, and we know that it's broken down into subdivisions. So the first division would be fungi. And fungi, again, are eukaryotes. Their cell walls are made of chitin, so they have chitin cell walls. They use organic chemicals for energy. Mold and mushrooms are multicellular, consisting of masses of mycelia, which are composed of filaments called hyphae. So if you look at this picture here of this plate, you'll see that you have this great mold growing on there, and you can see these little filaments, the little hyphae, and that forms this large mass called mycelia. Okay, and that's how mold grows. Molds are very, very big, as you can see. So you can actually see that with your actual eye. You don't even need a microscope to be able to see mold. Yeast are unicellular fungi. So similar to bacteria with one cell, whereas molds are multicellular. And remember that, again, molds are filamentous, so you do have filamentous molds. They reproduce sexually or asexually, so with a mate or without a mate. Most fungi are scavengers because they decompose dead organisms. Protozoa, the next subdivision, are eukaryotes. They are unicellular. They absorb or ingest organic chemicals. They may be motile via pseudopods, cilia, or flagella, and they also reproduce sexually or asexually. Next subdivision is algae, and they are also eukaryotes. They are unicellular, and they have cellulose cell walls. So can you remember at this point what the bacterial cell wall was made of? So the bacterial cell wall was made up of peptidoglycan. How about the fungi cell wall? It was made of chitin. Very good. Algae uses photosynthesis for energy, so it's photosynthetic. So if you ever had a fish tank at home and you left the light on all the time, you would notice that the tank started growing a green around it. That's algae. They use the light to grow. It produces molecular oxygen and organic compounds. So algae is good for fishes. It's just not good for us because we can't see it. We can't see the fishes that we have in our tank. And they also have asexual and asexual forms. The last group within the eukarya are your animals. And in microbiology, we talk about the multicellular animal parasites. They are also eukaryote, and they are our multicellular animals. They have no cell walls, so when we talk about them in microbiology, we're talking about parasitic flatworms and roundworms called helminths. They have microscopic stages in their life cycles. They go through sexual reproduction, and motility is either absent or reduced. So some examples of some multicellular animal parasites would be tapeworms. So I'm sure you heard of tapeworm before. Very prevalent in those little slider turtles that kids like to play with. So it's very important that they wash their hands before they touch after they finish playing with these turtles. And another example is I know some of us have puppies or doggies at home, and every month we're supposed to give them heartworm medicine. And we do have some some animal owners that just don't believe in giving their dogs heartworm medicine, and sometimes this will happen. And what the dog does acquire heartworm, and it just eats right through its heart. Viruses. Remember we said that viruses do not fit into the 3 domains. Why? Because viruses are considered to be acellular. They're not an actual cell. When you look at a virus, it consists of a DNA or RNA core. It has a core that is surrounded by a protein coat. That coat may be enclosed in a lipid envelope. Viruses are replicated only when they are in a living host cell. So they're what we call obligate intracellular parasites. They have to be inside of a host in order for them to be replicated, in order for you to make more of them. So if they're on the door handle, if they're in your hand, if they're on your steering wheel, they're not going to cause any infection because they are acellular. However, once they get inside of the human body, then they can cause problems. Here ends part A of chapter 1.